We're very pleased to be joined today by Sonja Kotz. She holds a chair in neurophysiology, neuropsychology and translational cognitive neuroscience at the Faculty for Psychology and Neuroscience and the Department of Neuropsychology and Psychopharmacology at Maastricht University. She's led multiple prestigious academic societies, served on a long list of scientific advisory boards, and is editor for multiple major journals. Her research started at the Max Planck Institute, where she was investigating how predictive cues facilitate the perception and cognition of verbal and nonverbal speech, language, and communication. At University of Manchester, she extended the research to the analyses of neural oscillations and computational modeling to understand how learning mechanisms apply to speech, language, and music. And now she's working to specify the neural network interfaces of frontostriatal and cortico-cerebellar circuitries involved in processing and learning, with a particular focus on temporal, rhythmic, and emotional stimulus properties and contexts. She's working to substantiate her findings with animal models and also working to develop neuromodulatory therapies in persons with Parkinson's disease, stroke, tinnitus, and and she collaborates widely with everyone who is lucky enough to get to collaborate with her. And she's really led the way and made the pivotal discoveries for many of us who are interested in researching time and prediction in the brain. Yes, many of us are looking to follow in her footsteps and follow up on her really exciting work. So I'm very much looking forward to hearing what she's got to say now on the evolution of time and rhythm in perception and cognition.
Thanks so much, Jonathan. Very, very kind of you. But I think nowadays it goes both ways, right? The young and dynamic people on board of this fantastic topic give us a lot of insight and push us to our own boundaries. And I think that's part of exciting science. So I'm quite grateful for that as well. So I probably should have changed that title a tiny bit, because I have not the biggest plan to talk about evolution, but to kind of only target that in the realm of why the cerebellum in particular seems to be a critical brain area that we should consider more closely when we are interested in time and rhythm, but more so how it actually helps us to regulate time and rhythm in the broader context of perception, action, and cognition. That's, oh dear, here we go. All right. So, I mean, I like to start talking about the fact that time and rhythm is something that is integral to everything that we do in life. So if we walk, we have a certain step frequency. If we are in more complex situations like these synchronous swimmers, we already try to combine sound with movement. So that's when we start talking about entrainment in the classical sense of auditory motor coupling. But it also plays a role when we're just listening to sound and the environment in general. When we again try to bring this into a bigger context, like in a choir, where we have to align not only our vocalization when we sing, but at the same time try to temporally align with other people so that the music that is generated comes across as coherent. And then we have speech and communication. In many ways, people have wanted to argue for the longest time that speech is arrhythmic and consequently doesn't necessarily benefit from an idea of rhythmicity. And in the case of communication, of course, we're looking at asynchrony, because if we all talk at the same time, speech becomes unintelligible, or we may even socially feel it's rude. These very complex ideas, so how we actually go from simple temporal information to more complex temporal information, and from there to rhythm, is of course something that we can study not only in the context. Can you actually see my cursor, if I may ask quickly?
Yes, we can.
Okay, super. So that is not just something that we want to look at in humans and across the lifespan, but we can also explore that in animal models. And these are the animal models that we're currently working with. So on the one hand, the rat, the macaque, and the seal, together with colleagues in the field like Hugo Marchand on the macaque work and the seals with Andrea Raviani. I like to also discuss something that I find somewhat opposing views in the field, and I put this right up there. So they're the more traditional views that I also tend to align with, and that is that there are two circuitries in the brain. On the one hand, the cerebellar circuitry, which you see marked here by 9, and the basal ganglia circuitry. And in the broadest of all definitions, these two systems seem to be complementary because on the one hand, they have different timescale sensitivities. So faster timescales related to cerebellar circuitry and slower timescales related to the basal ganglia. There's also the differentiation between interval-based and beat-based timing, respectively, for the latter for the basal ganglia. But there are also ideas that the one system is more processing temporal information almost without awareness, and that is the cerebellar system, while the basal ganglia seems to be really, really engaging in attentional processing of temporal information. So the biggest question is, why do we need this subcortical cortical interface to process temporal information, whether it's low-level temporal information or high-level temporal information, like we find it in speech and music. And one idea is indeed that these are critical structures in learning on the one hand, and on the other hand, in monitoring the outcome of cortical representations and their efficiency. So you can think about it from a predictive coding perspective, namely, we have expectations about when something occurs in a signal, but of course also what may occur next. And these systems may be critically involved with their differential cognitive engagement to monitor the matching between what is expected and what is actually happening. And consequently, they're constantly active, but we don't always see them, in particular when we scan. Alternatively, of course, we have the idea of chronomaps, and that is beautiful work of Dominica Buti. And here the idea is much more that we have these maps distributed across the cortex, and they're specific to durations on the one hand, but maybe are also driven by particular task demands in how we engage with this temporal information. This means the subcortex doesn't really necessarily play a role. Just to say that there are these two models out there at the moment, and we can think about that in the realm of the data that I will present to you today. So what I will do is I will present you data with the measures that we actually use in the lab. So on the one hand, we work with EEG and do classical event-related brain potential work, but we also work a lot in the oscillatory domain. So we do frequency tagging, rhythmic tracking, and phase angle analysis of oscillations. We do SASCPs, and we use also tracking data in the MRI. And the reason why I put it up here today is because I'm going to talk about one data set. So I want to give you the framework for that. All right. So the way I have built up my talk is that I will go through the facets of temporal information, going from very, very small temporal scale to temporal complexities in speech and music. just to walk you through the contribution of these subcortical structures. And on the one hand, I'm working comparatively, so we have animal models, but I also work translationally, which means I work a lot with patient data that can give us at least indirectly causal evidence for the contribution of particular brain regions in that circuitry to kind of prove the point why these different types of circuitries are critical when we process temporal information. So I'd like to start out with some work, more recent work that allows us to look into the facets of temporal order and probing the circuitry in particular. And I'm going to focus into the role of the thalamus, because at the moment, also from our conceptual thinking, the thalamus seems to be a critical connecting point between these two subcortical circuitries, the basal ganglia and the cerebellar circuitry. And it's going to be important to understand whether, in particular, subcompartmentalization of the thalamus in the auditory domain is contributing to processing stimuli that are not just content predictable, but also temporally predictable. And that is what you see at the top here. So we work with a classical oddball paradigm that we often use, and we have two temporal contexts. One is the regular timing context, and the other one, the irregular timing context. We have what we call a standard and a deviant pairing, but it can also be standards and standards. And part of the reason why we use these repetitions of standards or deviants is that we're interested in trying to understand the thalamus' role in sensory gating of information, which is particularly critical in the context of some clinical profiles such as tinnitus. And that's the model that I will be presenting here in this context. And then, of course, you have multiple ways to look at these timing manipulations, but the long and the short of it is that we normally expect facilitation effects of detecting a deviant in a temporally predictable context on the one hand, and we also expect a facilitation or a reduction effect when a stimulus is repeated. This is the classical sensory gating idea. So the question was, what role does the thalamus play in timing and in its function as a cortico-subcortical interface? So on the one hand, we have this rat model, and what we do here is it's a tinnitus rat model. So the animals are either noise exposed or not noise exposed to kind of mimic what tinnitus would actually be all about. And then they're implanted with deep electrodes in the mediate geniculate body of the thalamus. In parallel, we do similar kinds of studies now with patients, human patients who suffer from very persistent and excruciating tinnitus. And undergo deep brain stimulation in the very same structure. So the animal model is also serving as a clinical piece of evidence for the efficiency of this approach in tinnitus patients. And I'm going to show you some additional data on lesion symptom mapping that kind of combines the cortico-thalamo-cortical cerebellar interface, but one step at a time. So what I'm showing you here in a schema is first where the electrodes are implanted in the rat, and you see across a number of different schematics here, the placement of the electrodes in the mediate geniculate body. Here you see a staining thereof. So here you see the electrode placement. And I can already show you right away that these are now what we call local field potential responses in the mediate geniculate body of the rat brain in response to standard tones and deviant tones, standard 1, standard 2, deviant 1, deviant 2. So what we very clearly see here is a response to deviance and a reduction in the response to the second stimulus, so the gating effect, both for standards and deviance. By the way, this is a recording of a non-noise exposed rat, because we need the comparison between noise exposed and not noise exposed in order to look at the effect of whether DBS in the rat is actually leading to an improvement. So when I summarize this for you graphically, so this is the amplitude difference between the standard and the deviant tone. So we can replicate what we normally see at surface electrodes in humans. We see also an effect of stimulus position. So the second stimulus leads to a less increased amplitude than the first stimulus. And now we can look at the impact of noise exposure on the right-hand side and non-noise exposure on the left-hand side. And we can look at this as a function of irregular and regular timing, irregular in gray and regular in white. And we actually see that before deep brain stimulation, there is a difference in the non-noise exposed animals. So this would be a normal hearing animal. And the difference increases, as a matter of fact, when they then are deep brain stimulated. But we don't see a difference in timing benefit in those animals that have been previously noise exposed, but that changes after they receive deep brain stimulation. So what can we deduct from this? This tells us that this model, however indirect it is, serves the purpose of showing that the thalamus is not just regulating frequency modulations, but also is responsive to temporal manipulations that can be critically affected in clinical profiles such as tinnitus. And what I'm showing you now is the result, and we're really at the beginning of analyzing these data from a total of 4 patients who have undergone deep brain stimulation in the medial genicular body, to look at the correspondence of measurements from the surface electrodes that we measure from in these patients and from their local field potentials in the medial genicular body of the thalamus. And I'm giving you the example of one participant. So what we can very clearly see is very early on, this, by the way, are just the analysis for the standard tones and in the regular conditions. So not yet the full display as we've seen it in the rat model. And we see very nicely that we see very early on in the thalamus a positive peak that is much earlier than what we normally see at the surface, namely 50 milliseconds to P50, which is involved in sensory gating. But we see also a response a little bit later at the cortical surface. Later on, when we move towards the classical responses in the first 100 milliseconds, so we see a negative response in the thalamus that is, again, earlier than for the response at the cortical level. But you may see that there is some modulation earlier on that may or may not align with what we see subcortically. So this starts to become a rather interesting dynamic between the feed forward and potentially feed backward modulation of temporally predictable sound between the surface and the thalamus. And we see some modulation between the participants. At the moment, we have 4. But we see overall always a bit of a delay at the cortical level relative to the subcortical level. Part of the reason why this may be variable, this is like a schematic to show you the implantation of the electrodes in the patients. So they get implanted in the left and the right medial geniculate body, and you see that the alignment is not perfect, but as good as it gets when you actually do these types of invasive deep brain stimulations in patients. So more to come along the lines of that in the near future. So now we have kind of established that the thalamus is not just critical for frequency modulations, but also for temporal information processing. But the thalamus is not just a floating structure, it's a connecting structure between the cortex and subcortical structures. So one question that we were actually pursuing very actively is, can we get to data that shows to us that there is structural connectivity, for example, between the cerebellum and cortical structures that would involve the thalamus along the way, that would allow us to actually say there is a structural reality to this temporal information processing between the cerebellum and the cortex, to actually demonstrate that this is to be reckoned with. And what we did here is the following. We had patients, patients who had lesions in the posterior superior temporal gyrus of the left hemisphere, sparing the primary auditory cortex. And they underwent a number of tests going from threshold discrimination of order and micropatterns to then phoneme discrimination, pseudo-word discrimination, and word discrimination with variable accentuations for those of you who understand the linguistic aspects of place, manner, and voice. Which is not the most critical part. The most critical part is that there were patients who had discrimination problems across the board. So not just for temporal discrimination, but this type of discrimination also translated into higher level speech signals. And we took the behavioral results of these patients and did then what we call lesion symptom mapping. So the symptom is the deficit, and then we zoom into where the lesions are maximally located, going along with these deficits. By the way, those are only the data of those patients who were more than 2 standard deviations from the mean. So there are also patients who didn't have consistent deficits across these different tasks, which we used as a control in our analysis. So when we reduce the symptom lesion mapping, we end up with this area in the posterior superior temporal gyrus or sulcus, which is then used as our seed region to map in now the healthy, aging, comparable brain, because we want to avoid looking at compensation through reorganization of white matter, for example, and track into the brain. And what you see here now is, again, the location of that seed. And the tracking at the cortical level is delivering us exactly what we should expect from that area in the posterior superior temporal lobe, namely the dorsal and the ventral pathway. And what you see here slightly covered by these two pathways is the connectivity into the cerebellum. And I zoom into that for you in red. Now, a lot of people argue, yeah, this could be all cross-fiber tracking and all sorts of complications with this DTI approach. So what we did is 2 types of control analysis. For the first one, we actually tracked from M1 into the cerebellum, and you see that there's some overlap, but there's a differentiation that comes across as anterior-posterior. And we tracked from those patients who did not have across-the-board deficits, and we could not reestablish the same type of connectivity that you see right here in red from this posterior seed region into the cerebellum. So what this means is, by means of this approach, we can actually show structural connectivity that goes along with certain behavioral deficits that include timing deficits. And we take that as evidence for the reality of the cerebellum contributing to these fine-scale temporal information types that we manipulated in the behavior of these patients. All right, so just to summarize this for you, I think the comparative and translational studies showed us that the thalamus, in particular the auditory thalamus, gates temporal regularity in audition, and that this connectivity allows us to say something about the extraction and representation of temporal structure via the temporal pontocerebellar connectivity. All right, now let's move on from simple tones to, is there a question? I just see.
I think we will wait
Okay, all right.
for questions until the end.
Okay, so now I'm going to show you some results from work with the macaques. So this is the collaboration with Hugo Merchant. So in this particular context, we looked into isochronous tone sequences again, with deviant tones in between, but here was a particular manipulation. So the tone sequences were of different length, and that also means we can have a look at a phenomenon that everyone knows, but we may or may not all engage the same way in this phenomenon. Namely, even though you may be listening to physically identical tones, besides the deviant tone, you may start doing very subtle grouping in a sense of strong, weak, strong, weak, strong, weak, or we call it also the TikTok phenomenon. And in the literature, it's also been called subjective accentuation. So indeed, if you do that by manipulating the length of the tone sequences, you can force the system to actually go into this grouping. For me, it was an interesting way forward to actually do studies with the macaques that have normally been tested with a lot of training and reward. And I said it would be interesting to know whether we actually see a similar kind of grouping phenomenon in the macaque as in the human, if they're simply exposed to these sound trains, because it would tell us something about their basic grouping capacity. So this is what we're looking at here. And I'm going to walk you from left to right. And the top panel, you see the results of the macaque study, and at the bottom, the study with the humans. And what we did in the first place is we looked whether both the macaque and the humans respond similarly to the frequency of the stimulus train. So do they even follow the stimulus train? And they, as a matter of fact, they do. And then we started looking into two phenomena. On the one hand, how do they actually track the potential accentuation that may subjectively arise, namely the strong weak over time? And on the other hand, do they actually show an anticipation of when the next tone will arise? Okay, so do they make use of anticipatory behavior, which is often found in the beta range when you start looking into oscillations? What I want to show you here under C is an interesting phenomenon. So for the monkey, you see that was their session 4. And we're looking at the phase angle. So the pre-stimulus time window, which you see here in light blue, in different sequences. And what you very nicely see is that the phase angle is not a consistent phenomenon across different types of sequences. But when you ultimately average these, you end up with a particular phase angle that, interestingly enough, is very comparable between the monkeys and the humans. So even though in different types of tone sequences, you see this variability, on average, you see a similar type of phenomenon with regards to anticipating tone onset. We then can also do mean vector length statistics, where we use a randomization of the sequences that we use relative to those that were experimentally used. And you then can look at the statistical differences between these two types, both for the monkey and the human. And you see, again, very comparable results. So I think these first comparative studies show us that we see cross-species similarities of internalized timing, rhythm tracking, and making use of these subjective accentuations. We're following this up at the moment with more larger perceptual groupings. So also here, evidence to come in the near future. I now want to move a bit more towards temporal information processing in speech. And I will also show you one data set where we worked again with patients. But there's something that I haven't really talked about, and that is when we go to more complex stimuli like speech. Of course, speech is affected by different speech rates. Not everyone speaks exactly the same way, so we have variability there. And it also makes a difference whether or not a speech is intelligible or not. So the question for me that arises now is, are these temporal predictions, so onsets, critical onsets in the speech signal that is highly variable, actually showing, and which brain areas are involved in showing this? And then we again use a lesion model with cerebellar lesion patients to mimic what this form of temporal prediction in speech can actually look like. And I will break this down for you once I get to the data. So let's quickly look at the way this was done. So this was a prior study by my colleague Benedict Zöfel while he was still working in the UK. And what they looked at was different kinds of speech rates, so 3 Hertz versus 2 Hertz. And what you see is a manipulation of either speech intelligible in orange or vocoded speech, which is unintelligible. So there was alteration of whether or not the signal was intelligible or non-intelligible in both one rate and another rate. The task of the participants was to detect a target change or the consistency of a target showing in one or the other rate block. He was initially actually interested in something very different, namely the phenomenon of an entrainment echo, because the big question in the field of oscillatory analysis, temporal or not, has been, do we simply look at evoked responses because the brain is tracking the frequency of the stimulation? Or is there actually a phenomenon that tells us that the brain makes active use of a particular stimulation rate? And then we should actually see the brain continuing to make use of that stimulation rate in time windows where there is a break. So just to tell you the background. And of course, that empty time window can also become interesting because it, to a certain extent, allows you to also think about temporal prediction. Because if there's a pause in the signal, you would still make assumptions when the next signal onset needs to come, because otherwise, speech becomes sort of fragmented, and that is not what we normally do when we speak. So that's the background story. So he then showed me at the sensor level this activation, in this posterior region. And he said, this is something that I haven't really seen before. What do you think? And I said, well, the only way we can find out is to source localize this. And this is exactly what we did here. So I'm going to walk you through this. You see here under D and E, the source localization of the intelligible speech against 0 and 16 channels, so intelligible against unintelligible in E. And what we see very clearly is a massive activation spot in the cerebellum. Okay, so not entirely off to translate this sensor space activation into the source space, which you see here. And then we try to actually trace the signal from the sound offset. So when the signal actually stopped into the pause between the signals to actually see whether we see an effect of rate and intelligibility in this empty space and whether or not this can be indicative of a form of temporal prediction. And this is what you see here on the right-hand side. So again, we break this down by intelligible and unintelligible. And what actually comes out highly significant is the left inferior frontal gyrus. So when we now do tracking from the cerebellum, so cerebellum this time as the seed region to these different cortical structures, the right IFG, the left superior temporal gyrus, and the right superior temporal gyrus, we see effects also for the left STG, but this was not statistically significant, so there's always a question of power. But there's a very, very strong directed connectivity between the cerebellar seed and the left inferior frontal gyrus. Our idea is that in this rhythmic intelligible speech, we see a strong entrainment echo or an effect of temporal prediction in the cerebellum, and that these rate-specific connectivity patterns between the cerebellum and the left inferior frontal gyrus may be indicative of updating event timing in speech signals. Again, this is something that we're currently pursuing further because there are quite a few manipulations that need to be done to prove that this is actually a sound result. But it was a quite promising result in the first place. So now I want to give you one more data set here, and that is sort of indirectly causal evidence from cerebellar pathology. So what we did to kind of pursue this idea of temporal prediction in the related to the cerebellum was to look at healthy controls and cerebellar patients. And they were exposed to, again, two timing conditions, temporally predictable and temporally unpredictable. The way we implemented that was to simply use a basic visual auditory paradigm where the visual cue was actually temporally predicting the auditory cue in condition predictable, and in the unpredictable condition, that was not possible. And what we now look at is, in the first place, for the healthy controls on the left and on the right, the cerebellar patients, the condition effect. So is there a beneficial effect of prediction? And what you're looking at here in the beta range before the stimulus onset and the theta range after stimulus onset. So we actually see very little condition effect in the cerebellum, either pre-stimulus or post-stimulus, okay? We can now look at this also as a function of group differences. So now we look at, sorry, that's a bit too fast. We see here the group difference for the condition effect, so the predictable versus the unpredictable. And now I'm breaking this down for you in the realm of looking where this actually happened, in light blue for the healthy controls and in purple for the cerebellar patients. And the story is quite simple. We don't see any high beta modulation for the cerebellar patients pre-stimulus nor do we see a similar kind of theta response post-stimulus, which would be expected for sound in the cerebellar patients. So what can we conclude? So the cerebellum seems to be quite critically involved in monitoring for speech rate and intelligibility, and very likely plays a role in predicting event onsets in the complex speech signal. In the more simple setup that we used in the cerebellar patients, we see that cerebellar patients do not make use of temporal predictions when they're actually dealing with audiovisual stimuli. So this is some first evidence that this may simply be a very strong role of the cerebellum in these simpler and more complex stimuli. I'm not sure how I'm doing for time, because I can also stop at any point in time now and leave time for question, Jonathan.
I think it's certainly up to you at this point.
I can continue, but I want to make sure we have time. So,
well, I mean, people will start signing off in 15 minutes. So if you want to leave plenty of time, we can start taking questions now.
Okay. So I will then jump out of the talk. I mean, there are other studies that kind of simply imply that timing is quintessential, and it does impact also. For example, here's an example in younger children. This is work together with Raina Gordon, that in individual differences in expressive grammar use actually it can be explained to neural responses in musical rhythms, so different types of beat levels. So the connectivity between basic timing and rhythm and these more complex linguistic functions in this case is quite evident, not only in patients, but also in development of younger children. And yeah, that is what I meant to say. This is what we're currently doing, just to give you a perspective. So we continue working with other species. These are actually brains of the seal, which we have now scanned at 9.4 T to go into the connectivity pathways in the seal brain. Why the seal? It's been an interesting model for the vocal learning, rhythmic capacity hypothesis that Jonathan also entertains together with Ani Patel. And we're also adding some peripheral measures into the equation when we start measuring EEG from the seal. Sorry, this is a mouse. This is a bad example, but here's the seal and some ongoing work that will be upcoming soon. Yep, so thanks to the fantastic team, as always. You never do things on your own, and it's very important to acknowledge this, including the funders, and thank you for your attention.
Thank you, Sonja. All right, let me open the floor for questions. I guess maybe, but before I do, I should mention what Effie posted in the chat, just to clarify. Effie mentioned that the Timing Research Forum conference is open for registration. She just wanted to clarify that abstract submission is closed. So I just wanted to mention that. Okay, so anyone have questions for Sonja? I think this is a fantastic opportunity, and I will jump in if nobody else does, but yes. I'll wait a moment. Well, I can get us started. So in your studies on rhythmic intelligible speech, it seems like you've got a neural substrate for dynamic attending. Is that right?
Yeah, that's a good one. Potentially, yes.
So do you having localized this source of possible prediction activity in EEG, do you get the sense
This was
that,
actually MEG work,
oh, sorry,
John. I'm
MEG,
sorry.
MEG,
No.
yes,
Yeah, I should
but
have said so.
are you, do you think that cerebellum is keeping time, or is it making predictions based on an ongoing timing signal that's kept somewhere else?
That's a very good question. I mean, on the one hand, we know from animal work that the cerebellum has time scale sensitivity. There's no doubt about that. There's even lateralization of time scale sensitivity with right cerebellum being interested in faster transitions and the left in slower transitions. On the other hand, I think from more classical ideas of forward modeling, I think one could make the speculation that it's maybe interested in making use of these timescales, but in favor of making predictions and monitoring if predictions are coherent or not. And that we have also in a larger program on understanding this functionality and whether this is a more universal principle, have studied in cerebellar lesion patients quite extensively. And the way things look at the moment, it looks in favor of a more universal computation that may or may not be specific to temporal prediction for specific timescales.
Yeah, I like drawing the forward modeling literature into it. I think it makes a lot of sense. Sophie, I see Sophie's hand is raised.
Hi, Sonja. Thanks for.
Hi, Sophie.
I have a question on the same result. So, do you think this particular directed connectivity between the cerebellum and the left IFG, is that speech specific, or would you think that's more attention, or like in other task contexts, would you expect the cerebellum to connect to different regions then, or?
I mean, by all means, this is the cerebellum. We do know the cerebellum connects to almost every cortical structure. So I would think that ultimately there is functional differentiation as to where it connects in particular kinds of contexts. I mean, of course, now it would be interesting to also work with music and explore that in a similar manner, preferably even in a direct comparison. But I can easily see that there is functional and consequently also regional differentiation. The question is it still universal in its computation, or does it then become functionally specific?
Do you have an answer?
From the patient data at the moment, it looks very universal. So we've tested unisensory, we tested auditory motor, we have tested audiovisual. It always ends up being the same effect. These patients don't make use of predictability. And so in that sense, it looks more like a computationally universal strategy based on the patient data. There's also some TMS work by Joachim Groß's group that points in a similar direction. But yeah, one needs to test that across modalities and across tasks to have a final confirmation for that.
Thank you. Very exciting.
Let's see. Well, I've also got some questions about the thalamic work. So one thing I was curious about in the LFP signal in the MGB, in the medial geniculate body, so stimulating changes the LFP, but doesn't the LFP usually represent the inputs to a region? Or is in this case, is it indicating the output from the thalamus?
I mean, it is input because it's a response to the stimulus, right?
Okay,
But
so
what maybe I can clarify this.
Yeah.
What we ultimately now have to do as a next step, and that is what we're currently doing, is to actually look at the time course and if that's directional from the thalamus to the cortex and from the cortex back to the thalamus, because it's a feed forward, feed backward system. But for that, we need to dig deeper. So we're in the middle of doing this at the moment. So it's a lot of processing work going on.
Okay, and to clarify what you think the mechanism is there, the idea is that if you're doing this stimulation on the thalamus, then that early signal it's able to gate more effectively based on predicted timing.
Exactly. That's the idea behind it. And as a matter of fact, it may not have become entirely clear from the data that I showed in the animal model. When the animal receives deep brain stimulation, it starts making use of the temporal information again, but it doesn't make use of the temporal information when it's just noise exposed. Okay, so which is a critical contribution to the current idea that yeah, the thalamus is doing tonotropy, and it mirrors the frequency modulation at the cortical level. But what we now can show is that this is actually also responsive to temporal information.
So there's some temporal signal that's coming into the thalamus in advance of the stimulus.
Exactly.
That then is controlling its gating.
Exactly.
Yeah, is just based on the function of the thalamus. Do you have any sense of what kind of signal would contribute to its gating? Gating could be neuromodulatory, or I mean, yeah,
I
what
mean,
kind of
in the
signal?
most classical sense, it's like focusing attention on the stimulus quality, and I would now say stimulus timing, right? Because what
Yeah,
you
I
want
see.
to do is filter out noise.
So this could
That's
be
sort of the classical idea.
an intermediary between temporal attention and the effective temporal attention.
Exactly.
Mediates temporal attention. Oh,
Exactly.
I see.
Yes.
Yeah, that makes sense. All right, I should continue to open the floor to give others a chance before I grill Sonja with more questions.
Is there someone who has left something on the chat, or
I
was that
don't
other stuff? I don't know.
see in the chat yet. Let's see.
Can I jump in again on
Yeah. Oh, please do.
on Jonathan's question? You always ask the questions that I also had. So about the thalamus. So, but where does the thalamus get the timing signal from?
Where?
Yeah, or how does it get it?
That's yet a big question, right? And I think the idea that we're currently entertaining is probably well known to auditory neuroscientists, right? That you have a nonlinear pathway and a linear pathway coming all the way from the cochlea and the brainstem into the system, and the nonlinear one would potentially enter very quickly the cerebellum, going from the cerebellum to the thalamus and then to the frontal cortex. And in some older literature, we have actually found, for my taste, a very interesting concept, namely the wake-up call to the cortex. So be prepared. I really like that idea. But yeah, one needs to look into the connectivity in a much more differentiated manner than what we have done so far in order to actually support this.
Like the dual pathway model through.
Yeah, people call it dual pathway, and then there's all sorts of different terminology, but yeah, I call it non-linear or linear pathway, and then the linear one would be the classical auditory pathway, going from the thalamus to the temporal cortex and then to the frontal cortex, while the non-linear one may go more directly from subcortical to thalamus to cortex, to simply prepare the cortex for potential change or relevant change. Yeah.
But so you would say it's a bottom-up signal. So if
Yeah,
there
the nonlinear one,
top-down
more
prediction
bottom up.
protect,
Yeah. Yeah.
But not a preparatory signal based on an ongoing timing thing. So it's like the dynamic attending rhythmic speech.
Well, I mean, that I cannot separate right
Yeah.
now. I
Yeah.
mean, it could fit, Jonathan, but one needs to think about how one tests this, right?
Mhm.
I mean, I think I would have to do a few more manipulations here in order to make sure. It's not always easy when you work with patients because there's a limited amount of time what you're allowed to do.
I don't imagine that any of the things you do are easy, though it's easy for me to ask questions about them.
No, but they're all good and relevant questions. But the question is, yeah, how do we get there?
Mhm.
Maybe through modeling. I don't know. Modeling is always a good idea to try that, right? Yeah.
All right. Well, we're close to the hour, so we can let folks go. I think I may stick around for a few minutes and just look back through my notes and think of one or two more questions. But
Sure, I can
everybody,
stay
thank
on
you
that.
so much for attending. We hope you'll join us at the Timing Research Forum conference in Tokyo coming up. See the mailing and the website for more information. And thank you again to Sonja for joining us.
Thank you.
Yeah, so right. I'm becoming increasingly preoccupied with cerebellum recently as well.
Yeah, I haven't been disengaged from it in a long time. Even
Yes.
though people keep telling me, why are you looking there? And why are you not looking there? It's
Yeah.
it's yeah.
I'm actually curious to get your input on something, unless of course if anyone else wants to ask something, but I have a question about a specific experiment while I've got you on the phone. Um, okay. So the cerebellum, of course, also plays this important role in eye blink conditioning, which is the may also be learned in these connections from granule cells to Purkinje cells that are used to make predictions based on other things, based on forward models, based on potentially based on timing as well. And but if the cerebellum is making rhythmic predictions based on some kind of underlying rhythmic time signal, as it seems to be in like the rhythmic intelligible speech condition, would you expect to be able to learn eye blink reflexes based on rhythmic inputs that could then adjust by tempo? Because I'm planning an experiment like this, and I'm curious what you would think.
Yeah, it's a good one. I mean, there are multiple things that are interesting there, right? So the conditioning effect that's old stuff also from animal work that really clearly puts the cerebellum on the spot there. And but none of these studies have systematically varied rate or timing in that sense, right? They use a very statistical, and not even statistical, a very static setup. That's what I mean to say.
Yeah.
And people have not really explored that. So I think this is actually an interesting way forward to make systematic use of temporal manipulations.
Yeah,
And see how that affects.
Yeah, because it strikes me that like if the cerebellum is taking a rhythmic signal, something rhythmic happening in cortex or something, and learning connectivity from that to make very specific predictions, then it should be just as susceptible to, as long as that rhythmic signal is the most reliable cue, it should be able to learn the
Exactly.
reflexes based on that.
And I mean, that's the other thing. Both the basal ganglia system and the cerebellar system are quintessential in learning, and not just motor behavior or fine-tuning of motor behavior, but really also many other aspects. So I think these are the basic and most important questions, and I think it's one that I don't think I have seen in the field, but would certainly be worthwhile exploring because that would help to understand. And I mean, one could then think about it from the perspective that if there is this time scale sensitivity, that you can play with using the time scale sensitivity of the cerebellum relative to the basal ganglia, at least broadly speaking.
Right.
Because maybe it would not be interested in the time scale sensitivity of the basal ganglia, but more so of its own temporal dynamics.
Yeah, I still struggle with how to make sense of the long time scale sensitivity of the basal ganglia. Is it concatenation? Like I know
Yeah, I
that's
think it has a lot to do with building up sequencing or sequences.
uh-huh.
So and I mean, I know I find it so striking that the animal research has shown this very clear even lateralization of timescales for the cerebellum, but never for the basal ganglia, right? It's like, okay, the basal ganglia starts where the cerebellum stops. And then what? It goes days,
Yeah,
nights, months.
right.
I have
Because like
no idea.
if you're I imagine if you're counting seconds, that will show up the basal ganglia as sort
Yes.
of concatenation, and
Yeah.
maybe when we're not actually counting seconds, we're still doing something similar enough that that's how we keep longer.
I mean, there's still the reflection of whether the cerebellum is primarily driven by onsets, right? And doesn't really necessarily concatenate, while the basal ganglia is interested in making use of the onsets, but relative to the next onset. And
But you use
consequently, you
and
start.
using internal cues, I guess, as well.
Yes, yes,
So you could have the efference signal from an action rather than just the feedback
exactly.
to
Yeah,
initiate it,
yeah,
like internal counting.
yeah.
Um, oh wow, yeah, right. And I guess the cerebellum is just sitting at the end of these sensory pathways, especially audition and things that when it gets an onset signal, it's
Yeah,
ready to go.
I mean, we did play in the patients also with purely visual stimuli, and if you use similar kinds of timings as we used in the unisensory manipulations that we used, you see similar kinds of effects. So it's not modality specific as long as the timing is comparable, right? So, but then again, you know, we're probably more prone to think about time in the realm of auditory stimuli, unless the stimulus is dynamic. And part of the reason we used a dynamic stimuli also in the audiovisual paradigm was exactly that. I said, no, I don't want a static visual stimulus. I want a dynamic visual stimulus. And so as dynamic as the tone that follows, in a sense. So that was controlled.
You know this recent preprint from Divik and Narayan with the eye blink stimulation, and it's about like implicit timing of intervals.
The trace model, or is it more recent than
Yeah, well, it's like this year or end of 24.
Oh, okay, then I haven't seen that.
Yeah, it's quite nice results that the cerebellum like helps learn the distribution of the intervals at which the eye blink.
Right. Her Bayesian. So I know I remember that she had a paper on that, but I haven't seen. Here it is: cerebellar encoding of prior knowledge of temporal statistics.
Yeah, might be that.
Which one is? Can you share this by any chance on the chat?
That one looks to be in Bayesian, or sorry, on bioRxiv.
I think it's on bioRxiv. Yeah.
Yes.
Yeah, it's the Koppen is the first author. Yeah.
Well, thank you for that one.
Yeah,
Right.
she withered it as, and I was like, my worry was a little bit that because the eye blink conditioning is so aversive, so that the animal could just solve the task by trying to close the eye the earliest possible. So when the timing is more variable, it makes sense that you close your eye earlier to avoid the shock. But she has some control analysis in there where it's really more about really the temporal prediction. So it reflects the distribution and not just the
there it is.
earliest onset. Yeah. That's cool work.
We found the activity of the Purkinje cells changed with the temporal statistics. Optogenetic perturbation attenuated the predictive behavior, but left the eye blinks intact. Left reflexive eye blinks intact. And they've got a novel complex spike signal. Consistent with the onset of the earliest probable time interval. Oh yeah, yeah. Thank you, Sophie.
Did you share, Sophie?
Shall I share it? Sorry, I will.
Here's the link. One moment.
Here.
Zoom.
I'm losing.
There.
Oh, yeah. Okay. Yeah, that's it.
Here.
Sending that to my student now. my paper reader.
Oh, isn't that fantastic? You don't have bought into the cool age yet to use a notebook. What's it called? Notebook.
Yeah, paper reader. No, I'd prefer that whoever reads my paper for me learn something from it that they can use later.
Are you sure they're not using AI?
That's a good question.
That's what I'm trying to figure out. I mean,
course.
I've been quite puzzled how you can like upload this stuff, and we actually had a training session at one departmental meeting, and an expert, an AI expert, came and told us all about the wonderful use of AI, and then we all had a chance to try and explore it, and I was actually quite blown away, and I was like, then I got very irritated, and I was like, okay, so no one's ever going to read a paper anymore.
I know, it's so sad. Why am I doing good writing anymore?
Why even write it, you know?
Yeah. Yeah, but I'm worried that we're on a peak right now because we have trained these AI models on decades of human thought. And what's going to happen in 10 years when we train the AI models on their own output? So I don't know if it's going to continue to rise or if we're actually at a peak kind of will like at some point we just have
significantly.
Yeah.
That's
we stop producing all this output.
I think we need to turn the world into a big generative adversarial model where there are AIs that are reviewing the writing of other AIs and then selecting for good writing. And then
peer review system. Yeah. Okay.
Yeah.
As Russ Poldrack actually developed an AI tool to be a reviewer. So
no.
as a reviewer, and that was scary.
That is scary.
Yeah.
But he shares the code, so you can you can look at it.
Yeah.
Anyhow, I'm sidetracking. Sorry about that.
Very fast. Yeah.
I mean, in the end, this is a big community activity, and yeah, I mean, whether or not an AI reviewer would be better in some way, I'd love to get the AI help, but it's much more important that we be connecting with each other about this.
Yes, I agree. I agree.
All right. Well, I guess I can end the meeting. Sonja, thank you again for joining us for this. It's a pleasure and an honor to hear what you're up to now.
Yeah, I mean, not entirely new, at least for some people like Sophie.
But I mean,
yeah, but it helps to hear it multiple times. I understand new things every time I hear it. Actually, it's good.
Yeah, but lots happening at the moment. So
yeah, and actually, I've got I don't know, I could keep talking forever. I've got EEG results we're trying to publish that are in autism, and I've been very interested in the cerebellar differences in autism. And we're seeing less entrainment to prediction-related rhythmic stimuli in posterior electrodes in particular. And suddenly, and it hadn't even struck me to think that might be a cerebellar signal.
I mean, that's when, even though I trust MEG a lot more in terms of potential source mapping. I mean, I don't take surface electrodes as face value of localization, but that's why I said, okay, we need to go in, we need to look, and you can do that with MEG nicely. It's a bit more complex with EEG, but depending on the number of electrodes that you measure from, you can do source localization. So, but yeah, I found it quite striking to see that result, and I said, are you sure that's not an artifact? And yeah, we did a lot of work.
Yeah.
So
Well, because I know I've been looking at Neil Todd's couple of papers looking
yeah,
at cerebellar EEG and
yeah,
just wondering.
yeah. He has this new cap, right?
Yes,
So
exactly, the extension.
yeah, we wrote a grant together to see whether we can do more
Oh, really?
systematic testing of that cap. So yeah.
Well, I'm excited to hear about that. All right. I may pick one up myself.
Yeah, hey.
Just for the fashion statement. I feel like
The,
I could set a trend.
yeah, well, the more the merrier.
Yeah. Great. All right. Good to see you again.
Okay, good to see you.
Okay, thank
Take
you.
care. Bye-bye.
Bye.